Sure. Protozoans are structures of cells that are allowing the cells to protrude into another environment. And it's a way the cells have to communicate with other cells and also with the proteins that make the matrix of the tissues. An example are, for instance, cells of the bones, that they use this protrusion to digest the bone matrix and remodel afterwards by other cells. Or cells that we are studying that are on the immune system and use these protrusions to go in between cells of the other tissues and communicate also with the lumen of the guts, of the airways, and in this way making their space through other cells. It is challenging because they are made at one side of the cell, so at a certain surface. So if you want to study them by imaging, you need to have a substrate which is compatible with imaging. And what it makes also challenging is that they are multi-protein assembly. So they are made of many, many different proteins. It's highly crowded, and if you want to dissect each protein, you need to use several techniques, complementary techniques, to look at those. So that is pretty challenging, yeah. The advances in imaging techniques have been really fundamental for us to understand these structures, because we have to use different techniques to address different questions. Developments like super resolution microscopy, where you can look at extremely high resolved images of very crowded structures, have helped us to understand how these protozoans are structurally arranged, the architecture of these structures. And in this way we have also understood how the shape of this structure helps the function of these structures. And also development in the speed by which you can acquire images have been fundamental, because you can look, of course, much faster at the cellular processes that we are looking at. And these structures are extremely dynamic, so we wanted to look at the interaction with different molecules.